Welcome to Biology at Priyajit, my dear viewers. Today we will learn how RNA polymerase II in eukaryotes initiates transcription step by step. This enzyme makes messenger RNA or mRNA which carries the instruction from DNA to make proteins. We will explore how Pol II recognizes the core promoter, assembles with basal transcription factors and begin transcription process efficiently. By the end, you will understand how eukaryotic cells accurately convert DNA information into mRNA. Let's dive in and see how transcription kicks off inside the nucleoplasm. So location RNA polymerase II or Pol II works in the nucleoplasm of eukaryotic cells and it transcribes messenger RNA. It makes mRNA but cannot start transcription alone. It needs help from the basal transcription factors. So what is core promoter? The core promoter is the shortest DNA sequences where the RNA polymerase enzyme can bind and to start the transcription process. The core promoter basically contains the minimum signal for general transcription factors to assemble at the start site. Alone, it works very slowly, so activator proteins are needed for efficient transcription. Promoter proximal elements, they are located 100 base pair upstream of the start site. The activators bind here to increase the transcription efficiency, and these key sequences involve CAAT box which is located at the minus 75 base pair upstream, and GC box located minus 90 base pair upstream, and sequence is GGG CGG, and this sequence help enhance general transcription factor assembly. Initiation Initiated sequence or INR and TATA box. At the start point, mRNA usually begins with A flanked by pyrimidine, so it is PY2. There are two pyrimidine CA and there are 5 pyrimidine residues. So this is the sequence of the transcription. This is the CAT box, this is GC box, and this is the TATA box, and this is the octamer box. Many promoters also have a TATA box at minus 25 base pair upstream, also called name as Hogness box. This is the TATA box, also Hogness box, and TATA box is similar to minus 10 sequence in prokaryotic or bacterial promoter, but they located differently. Tata-less promoter, about 50% of promoter lack a Tata box. Instead, they may have a DPE or downstream promoter element that is located at 25 to 38. A core promoter can be Tata box plus INR or INR plus downstream promoter element. So now we should focus on the assembly of basal transcription apparatus. Transcription factors assemble step by step at the promoter site. TF2D binds first. It recognizes the TATA box. TF2 binds at the TATA box. It recognizes the TATA box via TBP or with the help of the TATA binding protein. TBP is supported by TAPs, TATA box associated factors. For TATA-less promoter, TAPs interact with the INR or downstream promoter element. Then, secondly, TF2A joins the initiation complex. It binds upstream the promoter. Then TF2B binds downstream of the TATA. It helps TF2F bind, which has the helicase activity, and RAP38 binding, which is like bacterial sigma factor like protein. Then TF2E joins downstream of RNA polymerase II, and it joins the initiation complex, and TF2F basically unwinding activity. We have mentioned that it has helicase activity. It joins the complex, and also RNA polymerase binds there. TF2E downstream of the binds and downstream of the RNA polymerase II, and ultimately TF2H and TF2I. TF2J, not I. The TF2H has also helicase activity. It phosphorylates the CTD tail of polymerase II and helps escape the promoter sequence, and it supports the elongation process further. So starting transcription, once Pol II is bound, phosphorylated bond formation starts. Some promoters may show abortive initiation, short RNAs made repeatedly, and the CTD phosphorylation releases transcription factors ending abortive initiation. Transcription then proceeds to elongation. Then closed to open complex is formed. Closed complex, here DNA is still double stranded, and open complexes where DNA unwinds at the start site. so that transcription can begin. So as we conclude, remember that eukaryotic transcription by RNA polymerase II is a highly coordinated process from core promoter recognition to the stepwise assembly of the basal transcription factors and finally to efficient initiation of mRNA synthesis. Every step is carefully regulated. Understanding this process not only strengthens your concepts for undergraduate, postgraduate and NET exams but also helps you appreciate how eukaryotic cells accurately convert DNA information into functional RNA. Keep revising, stay curious and continue exploring the fascinating world of molecular biology. Thank you.